The cell membrane, also known as the plasma membrane, is a fundamental structural component of all living cells. It serves as a selective barrier that separates the internal environment of the cell from the external surroundings. Composed of a phospholipid bilayer embedded with various proteins, the cell membrane plays a critical role in controlling the passage of substances in and out of the cell. To facilitate this controlled exchange, the cell membrane employs several mechanisms, including passive and active transport processes. Let's discuss all of them in detail. Diffusion is the process by which particles or molecules move from an area of higher concentration to an area of lower concentration. It is a fundamental concept in physics, chemistry, and biology and is responsible for the mixing of substances and the transport of materials in various systems. Diffusion occurs because of the random motion of particles, which is driven by the tendency of particles to achieve a state of equilibrium. In a system where there are areas with different concentrations of particles, the particles will move in such a way as to equalize the concentration throughout the system. This movement continues until the concentrations are roughly equal or until equilibrium is reached. Here's how diffusion happens. At the microscopic level, particles are constantly in motion due to their thermal energy. This motion is random, and particles move in all directions. Diffusion occurs when there is a concentration gradient, which means that there is a difference in the concentration of particles between two regions. Particles will naturally move from areas of higher concentration to areas of lower concentration, resulting in the spreading out or mixing of the particles. Eventually, as particles continue to move, they will become evenly distributed, and the concentration gradient will disappear. At this point, equilibrium is reached, and there is no net movement of particles. Diffusion can occur in gases, liquids, and solids. The rate of diffusion depends on several factors, including temperature, the size and mass of the particles, and the medium through which diffusion is occurring. For example, diffusion is faster at higher temperatures because particles have more kinetic energy, and they move more rapidly. Diffusion also occurs more quickly in gases compared to liquids and solids because the particles in gases are more widely spaced and have greater freedom of movement. Diffusion plays a crucial role in various biological processes, as it allows for the movement of molecules and ions within living organisms. Here are some examples of diffusion in biology. Osmosis is a specific type of diffusion involving water molecules. It's crucial for maintaining the balance of water inside and outside of cells. Water will move through cell membranes from areas of lower solute concentration to higher solute concentration until equilibrium is reached. Other than osmosis, there are a lot more applications of diffusion in human biology. In the human respiratory system, oxygen diffuses from the air in the lungs into the bloodstream, while carbon dioxide diffuses from the bloodstream into the lungs to be exhaled. This gas exchange is essential for providing oxygen to cells and removing waste carbon dioxide. Within cells, glucose and oxygen molecules diffuse into the mitochondria, where they undergo cellular respiration to produce energy, ATP, and carbon dioxide as a waste product. In the small intestine, the products of digestion, such as glucose and amino acids, diffuse from the lumen of the intestine into the bloodstream through the epithelial cells lining the intestine. This process is essential for nutrient absorption. In nerve cells, Neurotransmitters are released from vesicles in response to an electrical signal. These neurotransmitters diffuse across a synapse to bind with receptors on the receiving cell, transmitting nerve impulses. In the kidney, waste products like urea and excess ions diffuse from the blood into the renal tubules, where they are eventually excreted as urine. In plant cells, carbon dioxide diffuses into the chloroplasts where photosynthesis takes place. It is then converted into glucose, and oxygen is released as a byproduct, which diffuses out of the chloroplast and into the surrounding cellular environment. These examples illustrate how diffusion is a fundamental process in biology, facilitating the movement of essential substances in and out of cells, organs, and organisms, thereby maintaining the overall health and function of living systems. Facilitated diffusion is a passive transport process that allows certain molecules, particularly polar or charged ones, to cross the cell membrane with the help of specific transport proteins. Unlike simple diffusion, facilitated diffusion requires the presence of these proteins, as the molecules being transported cannot easily pass through the lipid bilayer due to their size, charge, or hydrophilic nature. Here are more details on facilitated diffusion. Two kinds of transport proteins are involved in facilitated diffusion: channel proteins and carrier proteins. Let's first talk about channel proteins. Channel proteins are integral membrane proteins that form channels or pores in the cell membrane. Channel proteins are highly specific to certain ions or molecules, allowing them to move through the channel. For example, aquaporins facilitate the rapid movement of water molecules, while ion channels like sodium channels and potassium channels allow the selective passage of specific ions. Now let's discuss carrier proteins. Carrier proteins bind to specific molecules on one side of the membrane and undergo a conformational change to transport the molecule to the other side of the membrane. The binding of the molecule induces the protein to change its shape, allowing the molecule to be transported. Once the molecule is released on the other side, the carrier protein returns to its original conformation and is ready to transport more molecules. An example is the glucose transporter. Facilitated diffusion is highly specific and selective. Each transport protein is designed to interact with specific molecules or ions, and they do not transport just any substance. This specificity ensures that only the necessary molecules are transported into or out of the cell. Facilitated diffusion does not require the expenditure of cellular energy. Instead, it relies on the natural concentration gradient of the molecule being transported. Molecules move from areas of higher concentration to areas of lower concentration, which is a spontaneous process. The rate of facilitated diffusion is limited by the number of available transport proteins. Once all the transport proteins are in use, the rate of diffusion plateaus, even if there is a higher concentration gradient. This is known as saturation. Let's discuss few examples of facilitated transport. Glucose is a polar molecule and cannot easily pass through the lipid bilayer. Cells use glucose transporters to facilitate the diffusion of glucose into the cell. Ion channels allow the facilitated diffusion of ions like sodium, potassium, calcium, and chloride across the cell membrane. Aquaporins are channel proteins that enable the facilitated diffusion of water molecules across the membrane. Facilitated diffusion is a vital process in maintaining the homeostasis of the cell by ensuring the controlled passage of specific molecules in response to the cell's needs and the surrounding conditions. Both of these processes, diffusion and facilitated diffusion, were examples of passive transport. Now let's talk about active transport. Active transport is a remarkable process that allows cells to move molecules against their natural flow, and it requires energy, usually in the form of ATP. So, let's break it down step by step. First, we have transport proteins. These specialized proteins embedded in the cell membrane act as the gatekeepers for active transport. Active transport is all about energy, and that energy comes from molecules like ATP. It's like the fuel that powers this process. The real magic of active transport is that it moves molecules against their concentration gradients. While diffusion naturally pushes molecules from high to low concentration, active transport goes the other way. There are different active transport proteins for various molecules. The sodium-potassium pump is a superstar, helping to maintain the balance of sodium and potassium ions for nerve impulses and muscle contraction. But it's not alone. There are many others in the cell membrane. There are two main types of active transport, primary and secondary. In primary active transport, energy from ATP directly powers the transport. In secondary active transport, the energy comes from ion gradients established by primary active transport. Active transport is essential for life. It's responsible for functions like nerve signaling, muscle contraction, nutrient uptake, and even stomach acid production. Imagine a world without it. Our bodies wouldn't function as they should. So, there you have it. Active transport, the process that defies nature's rules to keep our cells in perfect balance. Before proceeding further, let's look at the key differences between active and passive transport. In the world of cell biology, two essential processes govern the movement of substances through the cell membrane: active transport and passive transport. Active transport is like the energy-consuming superhero of cellular transportation. It requires energy, typically in the form of ATP. On the other hand, passive transport is the laid-back surfer of cellular transport. It does not require energy. Active transport moves molecules against their natural concentration gradient, from areas of lower concentration to areas of higher concentration, while passive transport lets molecules move with their natural concentration gradient from areas of higher concentration to areas of lower concentration. Examples of active transport include the sodium-potassium pump and the sodium-glucose cotransporter, while simple diffusion for small, nonpolar molecules and facilitated diffusion for larger, polar, or charged molecules are examples of passive diffusion. So, in summary, active transport is the energy-consuming process that moves molecules against their gradient, while passive transport is the energy-free process that goes with the flow of the concentration gradient. Along with active and passive transport, cell membranes also have bulk transport mechanisms. Bulk transport is a fundamental process that allows cells to move large quantities of materials across their cell membranes. Unlike simple diffusion or facilitated diffusion, which involve the movement of small molecules across the lipid bilayer, bulk transport mechanisms are responsible for the transportation of larger particles or significant amounts of substances. There are two primary types of bulk transport across the cell membrane: endocytosis and exocytosis. Let's first talk about endocytosis. Endocytosis is a fundamental cellular process that allows cells to take in substances from their external environment. This process involves the formation of vesicles by invaginating the cell's plasma membrane, enclosing extracellular material and transporting it into the cell's interior. There are several types of endocytosis. First, I will discuss phagocytosis. Cells engulf large particles like bacteria or cellular debris. Specialized immune cells, such as macrophages, use phagocytosis to help defend the body against pathogens. Next is pinocytosis. This is like cellular drinking. Cells engulf small droplets of extracellular fluid containing dissolved molecules, such as nutrients and ions. Last one is receptor-mediated endocytosis. This process is highly specific. Cell surface receptors bind to specific molecules called as ligands, and the receptor-ligand complex is internalized in clathrin-coated vesicles. The general process of endocytosis involves the following steps. The plasma membrane invaginates, forming a pocket around the target material. The pocket pinches off from the plasma membrane, forming a vesicle. The vesicle is often coated with proteins like clathrin in the case of clathrin-mediated endocytosis. The vesicle containing the internalized material is transported into the cell's interior. In the case of clathrin-coated vesicles, the clathrin coat is removed. Depending on the type of material taken up, the vesicle may fuse with early endosomes, late endosomes, or lysosomes. In some cases, the material is digested within the cell or processed for recycling. Endocytosis is essential for nutrient uptake, immune responses, and maintaining cellular homeostasis. It allows cells to adapt to changes in their environment and is crucial for various physiological functions. Now, let's talk about exocytosis. Exocytosis is a fundamental cellular process that involves the active transport of molecules, typically large and often membrane-bound vesicles, from within a cell to the cell's exterior. It allows cells to release a variety of substances, such as neurotransmitters, hormones, enzymes, and waste products. It is the opposite of endocytosis, where cells internalize substances through processes like phagocytosis or pinocytosis. Exocytosis, believe it or not, is a crucial event happening in your cells every day. Imagine your cells as bustling cities with tiny workers inside. In this city, we have three main characters. First, secretory vesicles. These are like delivery trucks filled with important cargo. Second, cell membrane or plasma membrane. Our city's border control, regulating what comes in and goes out. And the third one is cytoskeleton. Think of it as the city's roads, helping vesicles navigate to their destination. Exocytosis happens in several stages, just like a well-choreographed dance. Our delivery trucks, or secretory vesicles, are loaded with goodies like proteins and lipids. They cruise through the city streets, carried by special motor proteins along the cytoskeleton roads. The vesicles park at their designated spots, getting ready to make their special deliveries. It's showtime. The vesicle membrane fuses with the city border, which is the cell membrane, releasing its contents outside. The cell shares its cargo with the outside world, which can be neurotransmitters, hormones, or enzymes. The cell isn't wasteful. It quickly recovers its lost membrane through endocytosis, ensuring everything stays in balance. Exocytosis is a big deal in the cellular world. It's responsible for things like nerve cells communicate using neurotransmitters. Hormones regulate your body's functions. For example, digestive enzymes breaking down food in your stomach. Immune cells fight off invaders by releasing substances. Cleaning up the mess with waste product removal. So, there you have it, the exciting world of exocytosis. It's a bit like a carefully orchestrated performance that keeps your cells running smoothly and allows them to interact with the world around them. Before concluding the video, let's take a closer look at the fundamental distinctions that set endocytosis and exocytosis apart. Endocytosis, the first player in this cellular duo, is all about bringing stuff into the cell. It's like the cell's way of ingesting substances from its surroundings. Now, let's meet the second player, exocytosis. This process is all about exporting substances out of the cell. It's like the cell's way of shipping its products. There are different types of endocytosis, like phagocytosis, pinocytosis, and receptor-mediated endocytosis, while exocytosis has two main types, constitutive exocytosis and regulated exocytosis. Phagocytosis is the process of engulfing large particles or other cells, often for digestion. Pinocytosis is like the cell taking a sip, internalizing small droplets of fluid. Receptor-mediated endocytosis involves specific molecules binding to cell surface receptors and being taken in. Constitutive exocytosis involves the continuous release of substances, like waste and membrane components, without needing specific signals. Regulated exocytosis is controlled by signals or stimuli and is responsible for the precise release of specific substances, like neurotransmitters. The primary purpose of endocytosis is to help the cell with nutrient uptake, waste removal, and regulation of membrane composition. While exocytosis serves various purposes, including releasing hormones, neurotransmitters, enzymes, and other cellular products, as well as incorporating membrane proteins and lipids into the cell's outer layer. In a nutshell, endocytosis is the process of bringing substances into the cell, while exocytosis is all about expelling substances from the cell. Together, they play vital roles in maintaining the cell's environment, communication, and responses to external cues. To view more educational videos, do not forget to subscribe. You can also click the bell icon to receive notifications when new videos are posted.